So here's something worth thinking about as you settle in tonight. Every single living thing you have ever seen, every bird outside your window, every blade of grass pushing through a crack in the sidewalk, every dog that's ever licked your face, every person you've ever loved, every bacterium quietly living on your skin right now, all of it, every bit of it, runs on the same basic instruction system, the same molecular language, the same code. And that code is DNA, which you already knew, of course. Everyone's heard of DNA. But here's the thing. Most of us walk around with this vague sense that DNA is important without really understanding what it actually is, how it actually works, or why it's shaped the way it's shaped. And honestly, that's fine. You don't need to understand DNA to live your life. But tonight, if you're up for it, I want to take you through it slowly, piece by piece, because once you start to see how it all fits together, it's one of the most quietly astonishing stories in all of science. Not loud astonishing, not explosions and rocket launches. More like the feeling you get when you realize something enormous has been hiding in plain sight your whole life. And we're going to take our time. There's no quiz at the end. If you drift off somewhere in the middle, that's perfectly fine. That's kind of the point. But let's start at the very beginning, and I mean the real beginning, before anyone knew DNA existed, before anyone even had a clear idea of what a cell was. Back when the biggest question in biology wasn't how does heredity work, but something even more basic than that. The question was simply, what are living things made of? Because you have to remember, for most of human history, nobody had any idea. You could look at a leaf, you could look at a muscle, you could look at blood, and you just saw stuff. Continuous, smooth, undifferentiated stuff. The idea that all living matter was built from tiny individual units, what we now call cells, that idea didn't really take hold until the 1830s and 1840s. A German botanist named Matthias Schleiden looked at plant tissues under a microscope and said, "I think every plant is made of cells." And then his friend, a physiologist named Theodor Schwann, looked at animal tissues and said, "Same thing." Animals, too. Every living thing is built from these little compartments. That was a huge deal. It gave biology its first real organizing principle. But it also immediately raised a new question. Okay, so, living things are made of cells. But what's inside the cells? What's the stuff that makes them work? Now, microscopes in the mid-1800s were getting better, but they were still pretty limited. You could see the outline of a cell. You could see that there was a darker blob in the middle, which people started calling the nucleus, from the Latin word for kernel, like the kernel inside a nut. But what was in that nucleus? What was it doing? Nobody knew. And honestly, most scientists at the time weren't even asking the right questions yet. Because the whole idea of heredity, the idea that parents pass specific instructions to their offspring, was still incredibly murky. People knew that children looked like their parents. Obviously, farmers had been breeding animals and plants for thousands of years. But the mechanism, the actual physical thing that carried hereditary information from one generation to the next, that was a complete mystery. And this is where a young Swiss physician named Friedrich Miescher enters the story. And I love this part, because it's so unglamorous. It's 1869. Miescher is 25 years old, working in a lab in Tübingen, Germany, and he's interested in the chemistry of cells. Specifically, he wants to know what's inside white blood cells. And to get white blood cells, he needs pus. Actual pus. So he goes to the local surgical clinic and collects used bandages from patients recovering from surgery. Bandages soaked in pus. because pus is full of white blood cells. This is his research material. Used pus-soaked surgical bandages. Not exactly the romantic image of scientific discovery, right? But Miescher is meticulous. He washes the cells off the bandages, breaks them open with chemicals, and starts separating out the different substances inside. And he finds something unexpected. From the nuclei of these cells, he extracts a substance that doesn't behave like any known protein. It's rich in phosphorus, which is unusual. It's acidic. It doesn't dissolve the way proteins dissolve. It's something new. He calls it nuclein because it came from the nucleus. And that substance, that weird phosphorus-rich goo he pulled out of pus-soaked bandages in 1869, that's what we now call DNA, deoxyribonucleic acid. Miescher was literally holding the molecule of heredity in his hands. He just didn't know it yet. Nobody did. And here's the kind of tragic part. Miescher actually suspected that nuclein might have something to do with heredity. He wrote about it in letters. He had this intuition, but he could never prove it, and the scientific establishment wasn't ready to hear it. The prevailing assumption for decades after Miescher's discovery was that proteins were the important molecules in the cell. Proteins were complex. Proteins were diverse. Proteins clearly did things. Nuclein, whatever it was, seemed too simple to matter much. And that assumption, that proteins must be the carriers of hereditary information, persisted for an astonishingly long time. We're talking decades. Miescher died in 1895, never knowing what he'd found. And the molecule he discovered sat in a kind of scientific limbo for years. People studied it, sure. They figured out more about its chemistry. By the early 1900s, scientists had worked out that nuclein, now being called nucleic acid, was made up of smaller building blocks. A biochemist named Phoebus Levine, working at the Rockefeller Institute in New York, identified the key components. He showed that DNA was built from units he called nucleotides, and each nucleotide had 3 parts: a sugar, a phosphate group, and one of 4 nitrogen-containing bases. 4 bases, just 4: adenine, guanine, cytosine, and thymine. Those names might sound familiar. A, G, C, and T, the 4 letters of the genetic alphabet. Levine figured that out in the 1920s and early 1930s. But here's the irony. Levine's work actually made people take DNA less seriously as a candidate for the heredity molecule. Because Levine proposed something called the tetranucleotide hypothesis, which basically said that DNA was a boring, repetitive molecule, where the 4 bases just repeated in a simple, monotonous pattern, over and over, like a wallpaper pattern. A GCT, AGCT, AGCT. And if that were true, DNA couldn't possibly carry complex information. It would be like trying to write a novel using only one word repeated endlessly. So the scientific consensus hardened even further around proteins. Proteins were the smart molecules. DNA was just structural filler, maybe something that held the nucleus together, like scaffolding. Not important. Not interesting. Think about what that means. For roughly 70 years after Miescher first isolated DNA, the molecule that contains the instructions for building every living thing on Earth was dismissed as boring, as background material. And this happens in science more often than you'd think. Sometimes the most important thing is sitting right there on the lab bench and everyone walks past it because they've already decided it can't be what they're looking for. Now, while all of this biochemistry was slowly unfolding, something else was happening in a completely different corner of biology, and the 2 stories wouldn't fully converge for decades. This other story starts in a monastery garden in what is now the Czech Republic, and it starts with a monk named Gregor Mendel. You've heard of Mendel. Everyone's heard of Mendel. But let me tell you what actually happened, because the real story is stranger and sadder than the textbook version. Mendel was an Augustinian friar living in Brno, and in the 1850s and 1860s, he started doing something that nobody else was doing. He was crossing pea plants in the monastery garden and carefully, obsessively counting the results. Not just looking at them and making general observations, but actually counting. How many plants had purple flowers versus white flowers? How many had smooth peas versus wrinkled peas? How many were tall versus short? He tracked these numbers across multiple generations, thousands and thousands of individual plants, and what he found was remarkable. The traits didn't just blend together the way most people assumed. They followed precise mathematical ratios, 3-to-1, predictable, repeatable. And from these ratios, Mendel deduced that hereditary information must come in discrete units, what he called factors, that are passed from parents to offspring according to specific rules. Each parent contributes one factor for each trait. Some factors are dominant, some are recessive, and they sort independently of each other. This was, and I don't think it's an exaggeration to say this, one of the most important insights in the history of biology. Mendel had discovered the basic laws of inheritance. He published his results in 1866 in a paper that laid out the whole thing clearly and elegantly, and almost nobody noticed. The paper appeared in the Proceedings of the Natural History Society of Brno, which was not exactly a high-profile journal. Mendel sent copies to some prominent scientists. Most of them ignored it or didn't understand it. The famous botanist Carl Nageli corresponded with Mendel, but basically told him his results were interesting, but not convincing. Mendel eventually gave up on the pea experiments, got promoted to abbot of the monastery, spent his later years dealing with administrative headaches and a tax dispute with the government, and died in 1884 without ever knowing that he'd uncovered the fundamental rules of heredity. His work was rediscovered in 1900, independently by 3 different scientists, Hugo de Vries, Karl Correns, and Erich von Tschermak, all of whom found Mendel's paper while researching their own similar results. And suddenly, Mendel was famous. Posthumously, tragically, but famous. And now biology had a new framework. Heredity worked through discrete units, which people started calling genes, a term coined by the Danish botanist Wilhelm Johansen in 1909. But here's the question that immediately followed. Okay, genes exist. They follow Mendel's rules. But what are they made of? What is a gene, physically? What substance in the cell actually carries this information? And this is where the 2 threads of our story start to reach toward each other. On one side, you've got the biochemists who know that cells contain DNA and proteins and various other molecules. On the other side, you've got the geneticists who know that hereditary information comes in discrete units called genes. The question that would dominate biology for the first half of the 20th century was, which molecule is the gene? Is it protein? Or is it that boring, repetitive nucleic acid stuff that Levine said couldn't possibly carry information? The answer came in stages, and the first really compelling piece of evidence came from a gruesome experiment involving mice and pneumonia bacteria. In 1928, a British medical officer named Frederick Griffith was studying Streptococcus pneumonia, the bacterium that causes pneumonia. He was working with 2 strains. One strain was virulent, meaning it could kill mice. Under the microscope, these bacteria had smooth, glistening coats, so they were called the S strain, for smooth. The other strain was harmless. These bacteria had rough-looking coats, so they were called the R strain. Simple enough. Smooth kills mice, rough doesn't. Griffith did a series of experiments. He injected mice with the live S strain. They died. He injected mice with the live R strain. They lived. He took the S strain, killed it with heat, and injected the dead bacteria into mice. They lived because dead bacteria can't hurt you. All straightforward so far. But then he did something clever. He mixed the dead S strain bacteria with the live R strain bacteria and injected that mixture into mice. And the mice died, which should not have happened. The S bacteria were dead. The R bacteria were harmless. But somehow, the harmless R bacteria had been transformed into deadly S bacteria. Something from the dead, smooth bacteria had gotten into the living, rough bacteria and changed them permanently. Griffith called this the transforming principle. Something, some substance, had carried the instructions for making a smooth coat from the dead bacteria to the living ones. And whatever that substance was, it was carrying hereditary information, because the transformed bacteria passed their new smooth-coated, deadly nature onto their offspring. Griffith didn't know what the transforming principle was. He died in 1941 during the London Blitz, killed when a German bomb hit his laboratory. He never found out, but his experiment set off a hunt that would change everything. Because now the question wasn't abstract anymore. It wasn't just which molecule is the gene in some philosophical sense. It was concrete and specific. What is the transforming principle? What substance, physically and chemically, can carry hereditary instructions from one bacterium to another? And the person who would finally answer that question was already working on it, quietly, methodically, in a laboratory at the Rockefeller Institute in New York. His name was Oswald Avery, and he was one of the most careful, meticulous scientists who ever lived. And that meticulousness is exactly what the problem needed, but it's also part of why the story took so long to unfold, and why, even after Avery solved it, a lot of people still didn't believe him. Let me tell you a little about Avery, because he's one of those figures in science who deserves to be a household name but isn't. He was born in Halifax, Nova Scotia, in 1877, moved to New York as a kid, and spent almost his entire career at the Rockefeller Institute, which is now Rockefeller University. He was small, barely 5 feet tall, soft-spoken, a bachelor who lived for his work. Colleagues called him the Professor, or sometimes just Fess. He was the kind of person who would spend years on a single question, ruling out one possibility after another with painstaking experiments, never rushing to publish, never making claims he couldn't back up with overwhelming evidence. And he had been studying pneumococcus, the same bacterium Griffith used, for decades before Griffith's experiment even happened. So when the news of the transforming principle reached him, Avery was uniquely positioned to figure out what it was. He knew these bacteria inside and out. But here's the thing. Avery didn't jump on the problem immediately. He was skeptical at first, actually. The idea that you could permanently change the hereditary nature of a living organism by exposing it to some extract from dead cells, that was a pretty radical claim. Avery wanted to see it for himself, in his own lab, under controlled conditions. So he and his colleagues spent years, literally years, first reproducing Griffith's transformation in the test tube, outside of living mice, which was itself a significant achievement. They showed that you could take an extract from the S strain, add it to R strain bacteria in a glass dish, and watch the R bacteria transform into S bacteria. No mice needed. That meant the transforming principle was a chemical substance you could isolate and study. And then began the long, grinding process of figuring out what that substance was. Avery worked with 2 younger colleagues, Colin MacLeod, and Macklin McCarty. Together, they spent about a decade on this problem. Think about that. A decade. They weren't sequencing genomes with fancy machines. They were doing classical biochemistry, the hard way. They would take huge batches of pneumococcus bacteria, break them open, and then systematically purify the extract, stripping away one type of molecule at a time, testing after each step whether the remaining extract could still transform bacteria. If you removed the proteins and the extract still worked, then the transforming principle wasn't protein. If you removed the lipids and it still worked, it wasn't lipid. And so on. By 1944, they had their answer. They published a paper in the Journal of Experimental Medicine, Avery, MacLeod, and McCarty. And the conclusion was clear. The transforming principle was deoxyribonucleic acid, DNA. They had purified the extract down to essentially pure DNA. They had shown that protein-digesting enzymes didn't destroy the transforming activity. Lipid-removing treatments didn't destroy it. RNA-digesting enzymes didn't destroy it. But when they added an enzyme that specifically broke down DNA, the transforming activity vanished. The evidence was about as clean as you could ask for in 1944. And you might think this would have been the moment. The moment everyone said, "Oh, DNA is the molecule of heredity. Let's go figure out how it works." But that's not what happened. Not even close. And the reasons why are fascinating, because they tell you something important about how science actually works, which is messier and more human than people usually admit. The biggest reason for the skepticism was that old idea we talked about, the tetranucleotide hypothesis, Levine's notion that DNA was just a boring, repetitive molecule with the 4 bases repeating in a monotonous pattern. If DNA was that simple, how could it possibly carry complex genetic information? Proteins, on the other hand, were made of 20 different amino acids arranged in endlessly variable sequences. Proteins seemed like the obvious candidate for the molecule of heredity. They had the complexity. DNA didn't, or so everyone thought. There was also a more personal element. Avery was not a geneticist. He was an immunochemist, a bacteriologist. The geneticists of the era, the people who studied fruit flies and corn plants and thought about chromosomes and genes, they didn't always pay close attention to what the bacteriologists were doing. There were disciplinary walls, and some prominent scientists simply didn't believe that bacteria even had genes in the traditional sense. Bacteria were considered primitive, fundamentally different from plants and animals. So, a finding about heredity in bacteria didn't necessarily seem relevant to the bigger question of how heredity works in complex organisms. Avery himself was cautious in the paper. He didn't make sweeping claims. He presented the data carefully and let it speak for itself, which was very much his style. In a private letter to his brother Roy, he was more candid. He wrote something along the lines of, and I'm paraphrasing, "If we're right, and this is DNA, then it means that nucleic acids are not merely structurally important, but functionally active substances that can determine the biochemical activities and specific characteristics of cells." He knew what it meant. He just wasn't the kind of person to shout about it. So the 1944 paper was published. It was noticed. It was discussed. But it didn't immediately overturn the protein hypothesis. The scientific community needed more convincing. And that convincing would come, beautifully, from an experiment in 1952 that is so elegant, so simple in concept, that it's still taught in every introductory biology class today. This is the Hershey-Chase experiment, and it's worth slowing down for. Alfred Hershey and Martha Chase were working at Cold Spring Harbor Laboratory on Long Island, and they were studying bacteriophages, viruses that infect bacteria. Now here's why bacteriophages were so useful for this question. A bacteriophage is basically the simplest possible package of hereditary information. It's just a protein coat surrounding a core of DNA. That's it. 2 components, protein and DNA. When a phage infects a bacterium, it attaches to the outside, injects something into the cell, and that something hijacks the bacterium's machinery to make hundreds of new phages. So whatever the phage injects, whatever carries the instructions for making new phages, that's the hereditary material. And since the phage only has 2 candidates, protein or DNA, you just need to figure out which one goes inside the bacterium. Here's how Hershey and Chase did it, and this is the clever part. They used radioactive labeling. They grew one batch of phages in a medium containing radioactive phosphorus, phosphorus 32. Phosphorus is found in DNA, but not in proteins, so this labeled the DNA. They grew another batch in a medium containing radioactive sulfur, sulfur 35. Sulfur is found in proteins, but not in DNA, so this labeled the protein coats. Now they had 2 groups of phages, one with glowing DNA, one with glowing protein coats, metaphorically speaking. They let both groups of phages infect bacteria. They waited a few minutes for the phages to inject their material. And then, and this is the part that makes you smile, they used a kitchen blender, an actual wearing blender, the kind you'd make a milkshake in. They blended the mixture to shake the empty phage coats off the outside of the bacteria. Then they spun everything in a centrifuge. The heavier bacteria pelleted to the bottom. The lighter phage coats stayed in the liquid above. And then they checked. Where was the radioactivity? The sulfur 35, the protein label, was mostly in the liquid with the empty phage coats. It had stayed outside the bacteria. The phosphorus 32, the DNA label, was mostly in the pellet with the bacteria. It had gone inside. The DNA went in. The protein stayed out. And the DNA alone was sufficient to produce a whole new generation of phages inside those bacteria. That was it. DNA was the hereditary material, not protein. DNA. Published in 1952, and this time the scientific community listened, partly because the experiment was so clean and intuitive, partly because by 1952, the intellectual climate had shifted. Other evidence had been accumulating. Erwin Chargaff, a biochemist at Columbia University, had already dealt a fatal blow to the tetranucleotide hypothesis a few years earlier by showing that the 4 bases in DNA were not present in equal amounts, as Levine had assumed. Chargaff carefully measured the base composition of DNA from different organisms and found that it varied from species to species. DNA from a human had different proportions than DNA from a bacterium or a yeast. That meant DNA was not a boring, repetitive molecule. It had variability. It had complexity. It could, in principle, carry information. But Chargaff found something else, something even more interesting. And this is one of those details that seems like a footnote at first, but turns out to be a huge clue. He noticed that in every organism he studied, the amount of adenine was always roughly equal to the amount of thymine, and the amount of guanine was always roughly equal to the amount of cytosine. A equals T, G equals C, always. This became known as Chargaff's rules, and nobody quite knew what to make of them at first. Why would these particular bases always show up in matched pairs? What structural feature of DNA would produce that kind of regularity? The answer, of course, would come from the structure of DNA itself, and by the early 1950s, the race to figure out that structure was heating up. Multiple groups around the world were working on it, and the story of how the double helix was finally solved is one of the great dramas of 20th century science. It involves ambition, rivalry, brilliance, a critical piece of stolen data, and a young woman whose contribution was not properly recognized for decades. The key players were gathering. In Cambridge, England, a brash young American named James Watson had arrived at the Cavendish Laboratory, where he met a British physicist named Francis Crick, who was supposed to be working on his PhD thesis about protein structure, but kept getting distracted by DNA. In London, at King's College, a physical chemist named Rosalind Franklin was producing the best X-ray diffraction images of DNA anyone had ever seen, working alongside Maurice Wilkins, with whom she had a tense and difficult relationship from almost the very beginning. And across the Atlantic, at Caltech, the great Linus Pauling, arguably the most brilliant chemist alive, the man who had already figured out the alpha helix structure of proteins, was also turning his attention to DNA. It was early 1953, and the pieces were almost all on the table. Chargaff's rules, the X-ray data, the chemical knowledge of how nucleotides fit together. Someone was going to crack it soon. The question was who, and whether they would get the structure right on the first try. Because Linus Pauling, the genius, the favorite, had just published a proposed structure for DNA that was wrong, spectacularly wrong. And here's the thing. It wasn't wrong in some subtle, hard-to-spot way. It was wrong in a way that a good chemistry graduate student could have caught. Pauling proposed a triple helix, 3 strands wound around each other, with the phosphate groups, the backbone of DNA, stuffed into the center of the molecule and the bases sticking outward. And the critical error, the one that made chemists wince when they read the paper, was that Pauling had the phosphate groups unionized. At physiological pH, meaning at the conditions inside a living cell, those phosphates would absolutely be carrying a negative charge, every one of them. And if you jam a bunch of negatively charged groups into the tight center of a helix, they repel each other, violently. The structure would blow itself apart. It simply could not exist. Now, how does someone like Linus Pauling, a man who literally wrote the textbook on chemical bonding, make a mistake like that? That's a question historians have chewed on for decades. Part of it was probably overconfidence. Pauling had solved the alpha helix structure of proteins using model building and chemical intuition, and it had worked beautifully. He figured he could do the same thing with DNA. But he was also working at a disadvantage. He didn't have access to the best X-ray diffraction data that was being produced in London by Rosalind Franklin, and the U.S. State Department had actually confiscated his passport in 1952 because of his political activism, his peace advocacy during the early Cold War. So he couldn't travel to England to see the data firsthand or talk to the people who were closest to the problem. He was, in a sense, working half-blind. And when you're half-blind and you're Linus Pauling, you might figure your chemical intuition is good enough to fill in the gaps. This time, it wasn't. When Watson and Crick saw Pauling's paper, and they saw it before it was even officially published, because Peter Pauling, Linus's son, happened to be a graduate student at Cambridge and showed them a copy. They had 2 reactions. The first was relief. Pauling hadn't beaten them. The structure was wrong, and they knew it was wrong almost immediately. The second reaction was panic, because they knew Pauling would realize his mistake soon, and when he did, he would come back with the right answer. They figured they had maybe 6 weeks before Pauling corrected course. 6 weeks to solve the most important structural problem in biology. So let's talk about what Watson and Crick actually had to work with, because it's important to understand that they didn't discover the structure of DNA through experiments. Not directly. They didn't grow crystals. They didn't run X-ray machines. They didn't do chemistry at the bench. What they did was build models. Physical models made of metal and wire and clamps, like three-dimensional jigsaw puzzles, trying to find an arrangement of atoms that satisfied all the known constraints simultaneously. It's a method Pauling had pioneered, actually, and it requires you to hold a lot of information in your head at once: the bond angles, the bond lengths, the distances between atoms, the chemical rules about what can sit next to what. And then you need the experimental data to tell you the overall shape, the dimensions, the geometry of the molecule, so you know what your model has to fit inside. The experimental data they needed was coming from X-ray crystallography. And this is where we need to talk about Rosalind Franklin, because her story is central to everything that happened next. And it's a story that for a long time wasn't told properly. Rosalind Franklin was born in London in 1920 into a prominent Anglo-Jewish family. She was brilliant from an early age, precise, rigorous, not particularly interested in small talk or social niceties, deeply interested in getting things right. She studied natural sciences at Cambridge, then did important work during the war on the microstructure of coal, which might sound unglamorous, but was actually fascinating physical chemistry, and earned her a PhD. After the war, she spent several years in Paris, working in a crystallography lab, where she was happy, respected, and did excellent work. Then, in 1951, she moved to King's College London to apply X-ray diffraction techniques to biological fibers, specifically DNA. And almost immediately, things went wrong. Not scientifically. Scientifically, she was superb. What went wrong was the working relationship with Maurice Wilkins. The problem was partly a bureaucratic miscommunication. Wilkins, who had been working on DNA X-ray studies at King's, was away when Franklin was hired. He came back expecting her to be an assistant or a collaborator working under his direction. Franklin had been hired with the understanding that the DNA diffraction work would be hers, her own independent project. Neither of them was told clearly what the arrangement was supposed to be, and the resulting friction was immediate and lasting. Wilkins was reserved, somewhat passive-aggressive, and uncomfortable with confrontation. Franklin was direct, expected clarity, and didn't suffer ambiguity well. They were, temperamentally, almost perfectly designed to misunderstand each other. And the culture of King's College in the early 1950s didn't help. It was an institution where the senior common room was male only, where women were marginalized in ways both subtle and overt. Here's what matters most, though. Despite all of that, Franklin produced extraordinary work. She discovered that DNA existed in two forms. She called them the A form and the B form, depending on the humidity. The A form was crystalline and gave complex, detailed diffraction patterns. The B form was wetter, more extended, and gave a simpler but incredibly telling pattern. She was meticulous about separating these two forms and studying each one independently, which was exactly the right approach. And in May 1952, she and her graduate student Raymond Gosling took a photograph of the B form of DNA that became one of the most famous images in the history of science. It's known as Photo 51. If you've ever seen it reproduced, it's a pattern of dark spots and streaks arranged in an X shape, a characteristic cross pattern. To a trained crystallographer, that X pattern is essentially a signature. It screams helix. The spacing of the spots tells you the distance between repeating units along the helix. The overall pattern tells you about the diameter, the pitch, the symmetry, Franklin knew what she was looking at. In her notebooks, she recorded calculations that were moving her steadily toward the correct structure. She had the data. She was being careful, methodical, working through the mathematics rigorously rather than rushing to build models, which she considered premature. And this is where the story gets uncomfortable, because here's what happened. In January 1953, Maurice Wilkins showed photo 51 to James Watson, without Franklin's knowledge, without her permission. Watson visited King's College, and Wilkins, frustrated with his difficult relationship with Franklin, pulled out the photograph and showed it to him. Watson later wrote about the moment in his book, The Double Helix. He said that when he saw the photo, his mouth fell open and his pulse began to race. The cross pattern was unmistakable. DNA was a helix. And the measurements from the photograph gave critical parameters: the diameter of the helix, the spacing of the bases, the repeat distance of the structure. But that wasn't the only data that made its way to Cambridge without Franklin's knowledge. Max Perutz, who supervised Crick at the Cavendish Laboratory, gave Watson and Crick a Medical Research Council report that contained Franklin's detailed crystallographic calculations, her precise measurements of the unit cell dimensions, her analysis of the symmetry of the molecule. This was not published data. It was an internal report. And while Perutz later said he didn't think the report was confidential, the fact remains that Franklin's own quantitative analysis was shared with her competitors without her being aware of it. Think about what that means for a second. Franklin was doing the hard, experimental work, the painstaking, technically demanding crystallography that required real skill and patience. She was analyzing her own data carefully and systematically, and the key results of that work were handed to Watson and Crick, who used them as constraints for their model building. They knew the molecule was a helix. They knew its dimensions. They knew its symmetry. Now they just had to figure out how the pieces fit together. And even with all of that, they almost got it wrong. Watson's first attempt at a model had the bases on the outside of the helix, like Pauling's structure, with the backbone in the center. Crick pointed out problems with this. They tried pairing identical bases together, adenine with adenine, cytosine with cytosine, what's called a like-with-like model. That didn't work either, because the different bases have different sizes. The purines, adenine and guanine, are larger double-ringed molecules. The pyrimidines, cytosine and thymine, are smaller, single-ringed. If you pair two purines together, the pair is too wide. Two pyrimidines too narrow. The helix would bulge and pinch, and it wouldn't produce the regular, uniform structure that the X-ray data demanded. And then came the crucial insight. It came partly from a conversation with Jerry Donahue, an American crystallographer who happened to be sharing an office with Watson. Watson had been using the wrong chemical forms of the bases, the forms printed in most textbooks at the time, which turned out to be incorrect. Donahue told him to use the keto forms instead of the enol forms. This matters because the exact position of hydrogen atoms on the bases determines how they can form hydrogen bonds with each other. And when Watson started playing with cardboard cutouts of the bases in their correct chemical forms, he noticed something remarkable. An adenine in the keto form could form two hydrogen bonds with a thymine in the keto form. And the pair, the AT pair, was exactly the same width as a guanine-cytosine pair, which formed 3 hydrogen bonds. Every pair was one purine matched with one pyrimidine. Every pair was the same width. The helix would be perfectly uniform. And just like that, Chargaff's rules made sense. The reason adenine and thymine always appeared in equal amounts in any sample of DNA was because every adenine in the molecule was paired with a thymine on the opposite strand. Every guanine was paired with a cytosine. They weren't just floating around in equal proportions by coincidence. They were literally bonded to each other, face to face, rung after rung, like the steps of a twisted ladder. Watson reportedly came into the lab that morning and told everyone that he had found the secret of life, which was, not a modest claim. But he wasn't entirely wrong, because the base pairing rule didn't just explain the structure of DNA. It immediately suggested how DNA could copy itself. If you pull the two strands apart, each strand carries the information needed to rebuild the other. Wherever there's an A on one strand, you know there must be a T on the new strand. Wherever there's a G, you add a C. The molecule contains its own instructions for replication, built right into the way the bases pair up. Crick saw this immediately. In their famous paper, published in Nature on April 25, 1953, just a single page, maybe the most important single page in the history of biology, they included one of the great understated sentences in scientific literature. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That sentence is doing so much work. It's basically saying, we know this changes everything, but we're going to be cool about it. The structure of DNA doesn't just tell you what the molecule looks like. It tells you how heredity works at the molecular level. It tells you how a cell can duplicate its genetic information and pass it to daughter cells. It connects the abstract concept of a gene to a physical chemical reality, and it opens the door to understanding how that information might be read, how it might be translated into the proteins that actually build and run a living organism. But we're getting ahead of ourselves, because before we can talk about how DNA gets read and translated, we need to understand something about the structure itself in a bit more detail. Something about the way those two strands are oriented relative to each other, and why that orientation matters so much for everything that comes next. See, the two strands of the double helix don't run in the same direction. They run in opposite directions, and there's a specific chemical reason for that, and it creates a problem, a really elegant, interesting problem, for the cellular machinery that has to copy the DNA. So let's slow down here, because this is one of those details that sounds technical at first, but once you see it, it's actually beautiful, and it explains so much about how biology works at the molecular level. Each strand of DNA has a direction to it, like a one-way street, and the reason is chemistry. Remember, the backbone of each strand is made of alternating sugar and phosphate groups. Sugar, phosphate, on and on. The sugar in DNA is deoxyribose, and it's a ring-shaped molecule with 5 carbon atoms. Chemists number those carbons 1', 2', 3', 4', 5'. The prime just means it's on the sugar rather than on the base. It's a labeling convention. What matters is this. The phosphate group connects to the 5' carbon of one sugar, and the 3' carbon of the next sugar down the line. So the strand has a direction. One end has a free 5' group, and the other end has a free 3' group. We call them the 5' end and the 3' end. And here's the key thing. In the double helix, the two strands run in opposite directions. One strand goes 5' to 3' from top to bottom, and the other goes 3' to 5' from top to bottom. They're anti-parallel. Now, you might be thinking, okay, that's a nice detail, but why does it matter? And the answer is that it matters enormously because of how DNA gets copied. The enzyme that copies DNA, it's called DNA polymerase, can only work in one direction. It can only add new nucleotides to the 3' end of a growing strand. Only that direction. It reads the template strand from 3' to 5', and it builds the new strand from 5' to 3'. It's a one-way machine. It can't go backwards. And this creates a problem, because when you unzip the double helix to copy it, one strand is oriented in the convenient direction. The polymerase can just follow along smoothly, reading and building as the helix unwinds. That strand is called the leading strand, but the other strand is oriented the wrong way. The polymerase can't just cruise along it continuously because it would have to go in the direction the enzyme can't work. So what does the cell do? It copies that strand in short little chunks, working backwards in a sense, making a small piece, then jumping back, making another small piece, then jumping back again. These little fragments are called Okazaki fragments, named after Reiji and Tsuneko Okazaki, a husband and wife team working in Japan in the 1960s, who discovered them. That strand, the tricky one, is called the lagging strand. And then another enzyme comes along, DNA ligase, and glues all those little fragments together into one continuous strand. Think of it like this. Imagine you're painting a line on a road, and your paint machine can only move forward. On one side of the road, the direction you need to paint happens to be forward, so you just drive and paint. Easy. But on the other side, the line goes the other direction. So you have to keep stopping, picking up your machine, setting it down a little ways back, painting forward for a short stretch, then stopping again. Then someone comes along behind you and connects all those short stretches into one smooth line. That's basically what's happening at the replication fork. And this whole elegant, slightly awkward system exists because of that anti-parallel orientation, because the two strands run in opposite directions. Watson and Crick's structure predicted this problem before anyone even knew the machinery that would have to solve it. The structure came first, and then the biology had to be figured out. How does the cell actually deal with this architecture? And the answer turned out to be this beautiful, intricate choreography of enzymes, each with a specific job, all working together at the replication fork, moving at astonishing speed. In E. coli bacteria, the replication fork moves at about 1,000 nucleotides per second. 1,000 base pairs, copied, proofread, and stitched together, every single second. In your cells, it's slower, maybe 50 nucleotides per second. But you have thousands of replication forks working simultaneously across your genome. Your entire genome, all 3.2 billion base pairs of it, gets copied in a matter of hours, every time a cell divides. Let that settle for a moment. 3.2 billion letters, copied with an error rate of roughly 1 in a billion after proofreading and repair. That's extraordinary precision. And it all flows from the structure, from the base pairing, from the anti-parallel strands, from the chemistry of the sugar-phosphate backbone. But here's the thing. Knowing the structure of DNA, knowing that it could replicate, that was just the beginning. Because the really big question, the one that would consume molecular biology for the next decade and beyond, was this. How does the information in DNA actually get used? How does a sequence of A's, T's, G's, and C's tell a cell to make hemoglobin, or insulin, or keratin, or any of the thousands of proteins that a living organism needs? The structure tells you how the information is stored and copied, but it doesn't tell you how the information is read. This is where we need to talk about the genetic code. And the story of how the genetic code was cracked is one of the great intellectual adventures of the 20th century. It involved physicists turning to biology, mathematicians forming secret clubs, experimentalists racing each other with increasingly clever techniques, and a handful of truly wild ideas, some right, some spectacularly wrong, all converging over about 12 years into one of the most elegant solutions in all of science. Let's start with the basic problem. Proteins are made of amino acids, strung together in long chains. There are 20 different amino acids used in biological proteins. DNA has 4 different bases, A, T, G, C, so somehow a 4-letter alphabet has to specify a 20-word vocabulary. How? If each single base coded for 1 amino acid, you could only encode 4 amino acids. Not enough. If pairs of bases coded for amino acids, 2-letter combinations, you'd have 4 times 4, which is 16 possibilities. Still not enough for 20. But if you use 3-letter combinations, triplets, you get 4 times 4 times 4, which is 64. More than enough. So the hypothesis emerged pretty early that the code was probably a triplet code, 3 bases for each amino acid. These triplets would eventually be called codons. Now, one of the first people to think seriously about this was someone you might not expect, George Gamow, a theoretical physicist. Gamow was a big, gregarious, Russian-born physicist who had done important work in nuclear physics and cosmology. He was one of the architects of the Big Bang theory, and when he read Watson and Crick's paper in 1953, he got excited. He wrote to Watson and Crick directly, proposing an idea about how the bases in DNA might directly template amino acids through some kind of physical fitting. Like the amino acids would nestle into diamond-shaped holes between the bases in the double helix. It was wrong, completely wrong as it turned out, but it was creative, and it got people thinking about the coding problem in a rigorous mathematical way. Gamow even founded an informal group called the RNA Tie Club, 20 members, 1 for each amino acid, and they wore actual neckties with amino acid designs on them. The club included Watson, Crick, and a number of other luminaries. They circulated notes and papers among themselves, trying to crack the code. But here's what they were missing, what nobody fully understood yet. DNA doesn't directly make protein. There's an intermediary, and that intermediary is RNA, specifically messenger RNA. And understanding this required another set of discoveries, another set of experiments, another cast of characters. The idea that RNA might be involved in protein synthesis had been floating around for a while. Scientists had noticed that cells actively making lots of protein tended to have lots of RNA. The ribosomes, those little molecular machines where proteins actually get assembled, were known to contain RNA. But the exact role of RNA, the flow of information from DNA to RNA to protein, what Crick would eventually call the central dogma of molecular biology, that took time to work out. Crick articulated the central dogma in 1958, and it goes like this. Information flows from DNA to RNA to protein. DNA is the master copy, the archive. RNA is the working copy, the messenger that carries instructions from the nucleus out to the ribosomes. And protein is the final product, the functional molecule that actually does things in the cell. Information flows in that direction. It doesn't flow backward from protein to nucleic acid. Now, there are some exceptions and complications we know about today, reverse transcriptase in retroviruses being the most famous. But as a general principle, the central dogma holds remarkably well. So the picture that was emerging by the late 1950s was this. DNA gets transcribed into messenger RNA, and then messenger RNA gets translated into protein at the ribosome. Transcription and translation. 2 steps, and the genetic code is the dictionary that connects the two. It tells you which 3-letter codon in the messenger RNA corresponds to which amino acid in the protein. But actually cracking that dictionary, figuring out which codons specify which amino acids, that was a massive experimental challenge, and the breakthrough came from an unlikely source. In 1961, at a biochemistry conference at the International Congress in Moscow, a young, relatively unknown researcher from the National Institutes of Health named Marshall Nierenberg presented results that stunned the audience. Nierenberg and his colleague Heinrich Mattei had developed a cell-free system. Basically, they had ground up E. coli bacteria, removed the cell's own DNA and messenger RNA, and created a kind of molecular soup that still contained all the machinery needed to make proteins. Ribosomes, transfer RNAs, amino acids, energy molecules, everything was there, just waiting for instructions. And then they added their own synthetic messenger RNA. The first one they tried was the simplest possible message, a long string of nothing but uracil, poly-U. Remember, in RNA, uracil replaces thymine, so where DNA has A, T, G, C, RNA has A, U, G, C. So they fed this poly-U message into their cell-free system and asked, what protein comes out? The answer was a chain of nothing but phenylalanine, one amino acid, over and over, which meant that the codon U, 3 uracils in a row, codes for phenylalanine. That was the first word of the genetic code to be deciphered, and when Nierenberg presented this at Moscow, Crick himself was in the audience. Crick immediately recognized the significance and arranged for Nierenberg to present again to a much larger session. Overnight, Nierenberg went from an unknown to a central figure in molecular biology. He would eventually share the Nobel Prize in 1968 for this work. And then the race was on. Over the next 5 years, Nierenberg, Hargobind Khorana, Severo Ochoa, and others used increasingly sophisticated techniques to work out the entire genetic code, all 64 codons mapped to their amino acids. Khorana's contribution was particularly elegant. He figured out how to synthesize RNA molecules with repeating patterns, like UCUCUC or AAGAAGAAG, and then see what proteins they produced, which let him deduce the codons by logic. By 1966, the entire code was essentially complete, and it turned out to have some beautiful properties. It's redundant. Most amino acids are specified by more than one codon, since you have 64 codons but only 20 amino acids. 3 of the 64 codons don't code for any amino acid at all. They're stop signals, telling the ribosome to release the finished protein. And one codon, AUG, serves double duty as both the start signal and the code for the amino acid methionine. But here's what's really wild about the genetic code, and this is something worth sitting with for a moment. The code is essentially universal. The same codons specify the same amino acids in almost every living organism on Earth: bacteria, plants, fungi, insects, fish, you. There are a few minor variations in mitochondria and in some unusual organisms, but overwhelmingly, the code is the same everywhere, which means it was established very, very early in the history of life, probably close to 4 billion years ago, and it has been conserved ever since. Every living thing on this planet is reading the same molecular language. That's one of the most profound things biology has ever revealed. And it connects to something even deeper, something about how the information in DNA actually gets physically translated into the 3-dimensional shape of a protein, because it's the shape of a protein that determines what it does. The sequence of amino acids, determined by the sequence of codons in the messenger RNA, which is determined by the sequence of bases in the DNA. That sequence folds up into an intricate three-dimensional structure, and that structure is what gives the protein its function. An enzyme's active site, the shape of a hemoglobin molecule cradling an oxygen atom, the precise geometry of an antibody grabbing onto a virus. All of that comes from the folding, and the folding comes from the sequence, and the sequence comes from the DNA. It's this unbroken chain of information, from a linear code to a three-dimensional machine, and understanding how that folding works is actually one of the hardest problems in all of science. It's sometimes called the protein folding problem, and people have been wrestling with it for decades. Because here's the thing. If you know the sequence of amino acids in a protein, you know, in principle, everything you need to know to predict how it will fold. The instructions are all there, encoded in the chemistry of each amino acid, in how they attract or repel water, how they bond with each other, how they twist and bend under the influence of hydrogen bonds and van der Waals forces and all these subtle interactions. But the number of possible ways even a small protein could theoretically fold is astronomically large. A scientist named Cyrus Leventhal pointed this out back in 1969, and the thought experiment he proposed is still kind of staggering. If a protein tried to find its correct shape by randomly sampling every possible configuration, even a small protein of, say, 100 amino acids would need longer than the age of the universe to stumble onto the right one. And yet, in your body right now, proteins are folding into their correct shapes in milliseconds. So they're clearly not searching randomly. Something about the sequence guides the folding along a kind of energy landscape, funneling the chain toward its lowest energy state, its most stable configuration, through a series of intermediate steps. But predicting exactly what that final shape will be, just from the sequence alone, that turned out to be extraordinarily difficult. For a long time, this was one of those problems that humbled everyone who tried it. Christian Anfinsen won the Nobel Prize in 1972 for showing that the information needed to determine a protein's three-dimensional structure really is contained entirely in its amino acid sequence. He did this beautiful experiment with an enzyme called ribonuclease, where he unfolded it, denatured it, broke all its internal bonds, and then showed that it could spontaneously refold back into its correct, functional shape all by itself, no cellular machinery needed. Just the sequence, the chemistry, and the physics. So the answer was in there. But knowing the answer is in there, and actually being able to compute it, are two very different things. And for the next 50 years, computational biologists threw everything they had at this problem. Physics-based simulations, statistical methods, machine learning, massive computing power. And progress was real, but painfully slow. Then, in 2020, something happened that genuinely shocked the scientific community. A company called DeepMind, which is a subsidiary of Google's parent company Alphabet, entered a competition called CASP, the Critical Assessment of Protein Structure Prediction, which had been running every 2 years since 1994 as a kind of Olympics for protein folding prediction. And their system, called AlphaFold 2, essentially solved the problem. Not perfectly for every protein, but with an accuracy that was comparable to experimental methods like X-ray crystallography. The median accuracy was so high that many researchers described it as a paradigm shift. Something that had been one of biology's grand challenges for half a century was, in a meaningful sense, cracked. And it was cracked not by a deeper understanding of physics exactly, but by a neural network that had learned the patterns of protein folding from the database of known structures, which is its own kind of fascinating. The machine found regularities that human scientists had struggled to formalize. But let's not get too far ahead of ourselves, because there's a lot of ground to cover between the cracking of the genetic code in the 1960s and the era of computational biology. And one of the most important chapters in that story is about how scientists learned to actually manipulate DNA, to cut it, paste it, read it, copy it, and eventually rewrite it. Because understanding the code is one thing, being able to engineer with it is something else entirely, and it changed the world. So let's talk about restriction enzymes, because they're the scissors that made everything possible. In the late 1960s and early 1970s, researchers discovered that bacteria have a kind of immune system, not like your immune system with antibodies and T cells, but something much simpler and more ancient. Bacteria get attacked by viruses called bacteriophages, and to defend themselves, many bacteria produce enzymes that can recognize specific short sequences of DNA and cut the double helix at those points. These are restriction enzymes, sometimes called restriction endonucleases. The bacterium protects its own DNA from being cut by chemically modifying those same sequences in its own genome, adding little methyl groups that act like "don't cut here" flags. But any foreign DNA that enters the cell, like DNA from an invading virus, won't have those protective modifications, so it gets chopped up. It's elegant, right? A molecular defense system based on pattern recognition. Now, the reason this mattered so much for science is that these enzymes cut DNA at very specific, predictable sequences. An enzyme called EcoRI, for example, one of the first to be widely used, recognizes the 6-letter sequence GAATTC and cuts between the G and the A on both strands. And because the two strands of DNA are antiparallel, remember, the cuts end up being slightly offset, which leaves these little single-stranded overhangs on each end. They're called sticky ends because they can base pair with any other piece of DNA that was cut with the same enzyme, since the overhangs are complementary. Think about what that means. You can take DNA from any source, a human gene, a bacterial gene, a jellyfish gene, anything. Cut it with EcoRI, and the resulting fragments will have the same sticky ends. Mix those fragments with a bacterial plasmid that's been cut with the same enzyme, and the sticky ends will find each other. Base pair, and you can seal the joins with DNA ligase. You've just spliced foreign DNA into a bacterial chromosome. You've made recombinant DNA. This is the foundation of genetic engineering, and it came together remarkably quickly. In 1971 and 1972, Paul Berg at Stanford created the first recombinant DNA molecules by combining DNA from different organisms. In 1973, Herbert Boyer and Stanley Cohen took it further. They showed you could insert foreign DNA into a bacterial plasmid, put that plasmid into E. coli, and the bacteria would replicate it. The bacteria became little factories, copying the foreign DNA every time they divided. You could clone a gene. And almost immediately, people realized this was both incredibly powerful and potentially dangerous. There's a remarkable moment in the history of science that happened in February 1975 at a conference center called Asilomar on the coast of California. About 140 scientists gathered to discuss the risks of this new recombinant DNA technology. They were worried. What if you accidentally created a dangerous organism? What if you put a cancer gene into E. coli and it escaped the lab? The fact that the scientists themselves called for a moratorium on certain types of experiments and then organized a conference to establish safety guidelines That's almost unprecedented. Usually it's governments or the public pushing for regulation and scientists pushing back. Here, the scientists led. The Asilomar Conference resulted in a set of guidelines that shaped how recombinant DNA research was conducted for years afterward, with different levels of containment for different levels of perceived risk. It wasn't perfect, and there were heated arguments about whether the guidelines were too strict or not strict enough. But the fact that it happened at all says something important about the culture of molecular biology at that moment. And then the technology just accelerated. In the late 1970s, Frederick Sanger developed a method for reading the sequence of bases in a piece of DNA sequencing. His method, called chain termination sequencing, or the dideoxy method, was beautifully clever. The basic idea is that you set up a DNA replication reaction, but you spike it with modified nucleotides, dideoxynucleotides, that are missing a crucial hydroxyl group. When one of these modified nucleotides gets incorporated into the growing DNA strand, replication stops at that point because there's no hydroxyl group for the next nucleotide to attach to. You end up with a collection of fragments of different lengths, each one terminated at a different position, and by separating those fragments by size and reading which base is at the end of each one, you can reconstruct the sequence. Sanger used this method to sequence the entire genome of a bacteriophage called Phyx-174 in 1977. 5,386 bases, the first complete genome of any organism ever sequenced. He won his second Nobel Prize for this work. His second. The man had already won one for sequencing proteins. Around the same time, a completely different breakthrough was brewing, one that would eventually make almost everything else in molecular biology faster and easier. In 1983, a biochemist named Kary Mullis was driving along Highway 128 in Northern California, heading to his cabin in Mendocino County, and he had an idea. What if you could take a tiny amount of DNA and copy it over and over exponentially using a simple chemical reaction? The idea was to use two short primers, little pieces of synthetic DNA that would bracket the region you wanted to copy. You'd heat the DNA to separate the strands, cool it to let the primers bind, then use DNA polymerase to extend the primers and make new copies. Then you'd heat it again, separate everything, let the primers bind again, extend again. Each cycle would double the amount of DNA. After 20 cycles, you'd have about 1 million copies. After 30, 1 billion. He called it the polymerase chain reaction, PCR. Now, the initial version had a practical problem. The DNA polymerase they were using came from E. coli, and it got destroyed every time you heated the mixture to separate the strands. So you had to add fresh enzyme after every single cycle, which was tedious and expensive. The solution came from an unexpected place, the hot springs of Yellowstone National Park. Back in the late 1960s, a microbiologist named Thomas Brock had discovered bacteria living in the near boiling water of Yellowstone's thermal pools. One of these organisms, Thermus aquaticus, thrived at temperatures around 70 or 80 degrees Celsius, and its DNA polymerase, called Taq polymerase, was heat-stable. It could survive the strand separation step without being destroyed. Once Taq polymerase was incorporated into PCR, the whole process could be automated. Just set up the reaction, put it in a machine that cycles the temperature up and down, and walk away. Come back in a couple of hours and you've got billions of copies of your target DNA. PCR changed everything. Forensic science, medical diagnostics, evolutionary biology, archaeology, anywhere you needed to detect or amplify tiny amounts of DNA, PCR was the answer. Mullis won the Nobel Prize for it in 1993. And if you've ever had a COVID test, a real PCR test, not a rapid antigen test, you've personally benefited from an idea that a guy had while driving through the redwoods on a Friday night. So by the early 1990s, scientists had an extraordinary toolkit. They could cut DNA at specific sequences. They could paste pieces together. They could read the sequence. They could copy vanishingly small amounts into quantities large enough to work with. And all of these tools converged on what might be the most ambitious project in the history of biology. An effort to read the entire human genome. All 3 billion base pairs of it, letter by letter. It was called the Human Genome Project, and it officially launched in 1990 with a projected timeline of 15 years and a budget of roughly $3 billion. About $1 per base pair, which sounds almost poetic. The project was led initially by James Watson. Yes, the same Watson. Though he left in 1993 after clashing with the NIH director over the question of patenting gene sequences, and Francis Collins took over. And almost immediately, the project found itself in a race, because in 1998, a brash and controversial scientist named Craig Venter announced that his private company, Celera Genomics, would sequence the entire human genome faster and cheaper than the public project, using a different strategy. The public project was taking a methodical approach, mapping the genome first, breaking it into overlapping chunks, sequencing each chunk, then assembling the results. Venter proposed something called whole genome shotgun sequencing, where you just blast the entire genome into millions of tiny fragments, sequence them all, and then use powerful computers to figure out where they all overlapped and piece the whole thing together. Many established scientists said it couldn't work for a genome as large and repetitive as the human one. Venter said it could. And the rivalry that ensued was one of the most dramatic in modern science. It was personal, it was public, and it pushed both sides to work faster than anyone thought possible. Venter was a fascinating character. He'd served as a medic in Vietnam, came back, went to college on the GI Bill, and ended up at the National Institutes of Health, where he developed a technique for rapidly identifying genes using what he called expressed sequence tags. Essentially, little molecular bookmarks that flagged which stretches of DNA were actually being used to make proteins. He was impatient with the slow, methodical culture of government-funded science, and he had an ego that made him plenty of enemies. But he was also genuinely brilliant, and his whole genome shotgun idea, while risky, had a certain elegant audacity to it. Instead of painstakingly mapping the genome first, and then sequencing piece by piece, you just shatter the whole thing into fragments, read them all simultaneously using hundreds of automated sequencing machines, and then let the computers sort out the jigsaw puzzle. The key insight was that if you sequenced enough fragments with enough overlap, the overlapping regions would tell you how the pieces fit together. Think of it like tearing up 1,000 copies of the same book, reading random scraps from each copy, and then using a computer to match up the sentences that appear on multiple scraps until you've reconstructed the full text. The public project, led by Collins, responded to Venter's challenge by accelerating their own timeline dramatically. What had been planned as a 15-year effort suddenly became a race, and both sides poured resources into finishing first. The public consortium had the advantage of international collaboration, labs in the US, UK, Japan, France, Germany, and China all contributing, plus a commitment to releasing their data publicly every 24 hours, which meant anyone could use it. Celera had the advantage of speed, newer technology, and a single coordinated command structure. And honestly, Celera also quietly used some of the public project's freely available data to help with their own assembly, which was a source of considerable friction. The rivalry came to a politically brokered truce. In June of 2000, President Bill Clinton hosted a White House ceremony where both Venter and Collins stood together and jointly announced that a working draft of the human genome had been completed. Clinton compared the achievement to Meriwether Lewis unfolding a map of the American West for Thomas Jefferson. It was a feel-good moment, carefully staged to give both sides credit. The formal publications came out simultaneously in February 2001. The Public Consortium published in Nature, Celera published in Science. And the genome was, at last, laid open. Now, here's the thing that surprised almost everyone. The human genome turned out to contain far fewer genes than anyone expected. For years, the conventional wisdom had been that humans probably had around 100,000 genes, maybe more. We're complex organisms, right? Surely we need a lot of genes. But the actual count came in at somewhere around 20 to 25,000. That's barely more than a roundworm, which has about 20,000. A grape has about 30,000. A grain of rice has close to 40,000. So the sheer number of genes clearly wasn't what made humans complex. That was a genuinely humbling finding, and it forced biologists to rethink their entire framework for understanding how genomes work. The answer, it turned out, had a lot to do with regulation, not just what genes you have, but when and where and how much each gene gets turned on or off. And it had to do with alternative splicing, where a single gene can produce multiple different proteins depending on how its RNA transcript gets edited. So a relatively modest number of genes can generate a much larger repertoire of proteins. The complexity isn't in the parts list, it's in the choreography. And here's another thing that emerged from the Human Genome Project that really changed how people thought about DNA. Only about 1.5% to 2% of the human genome actually codes for proteins. That's it. The rest, the vast majority, was initially dismissed as junk DNA, a term that had been floating around since the 1970s. The assumption was that most of the genome was just evolutionary debris, old broken genes, viral sequences that had inserted themselves over millions of years, repetitive stretches that didn't do anything useful. And some of it genuinely is that. But over the years since the genome was sequenced, it's become increasingly clear that a lot of the so-called junk is actually doing important work. Some of it contains regulatory sequences that control when genes turn on and off. Some of it gets transcribed into RNA molecules that never become proteins but serve other functions. They help regulate gene expression. They play structural roles. They do things we're still figuring out. The ENCODE project, which launched in 2003 as a follow-up to the Human Genome Project, found that something like 80% of the genome shows signs of biochemical activity, though there's ongoing debate about how much of that activity is truly functional versus just biochemical noise. It's a genuinely open question and a fascinating one. The genome is not a tidy blueprint. It's more like a vast, messy, layered document that's been edited and annotated and scribbled on by billions of years of evolution, and we're still learning how to read it. But let's come back to what the Genome Project made possible in practical terms, because this is where it starts touching your life directly. Once you have the reference sequence of the human genome, you can start comparing it across individuals and asking, where do people differ? And it turns out that any two humans are about 99.9% identical in their DNA sequence. The differences, the 0.1%, amount to roughly 3 million positions scattered across the genome where one person might have an A and another person has a G or a C instead of a T. These single-letter variations are called single nucleotide polymorphisms, or SNPs, pronounced SNPs, and SNPs are incredibly useful. Some of them fall in or near genes and directly affect how those genes work. A SNP might change one amino acid in a protein, or alter how much of a protein gets made, or have no detectable effect at all. But by cataloging millions of SNPs across thousands of people and correlating them with traits and diseases, researchers have been able to identify genetic risk factors for conditions like diabetes, heart disease, Alzheimer's, various cancers, and hundreds of other things. This is the basis of genome-wide association studies, or GWAS, which became one of the dominant research strategies in genetics after the Genome Project. And this is also what's behind the consumer DNA testing kits you've probably seen advertised. When you spit into a tube and send it to a company like 23andMe, they're not sequencing your entire genome. That would still be relatively expensive, though the cost has plummeted. The first human genome cost about $3 billion, and today you can get a whole genome sequenced for under $1,000, sometimes a few hundred. But the consumer kits typically use a SNP chip, a small glass slide with hundreds of thousands of known SNP positions on it, and they check which variant you have at each one. From that, they can estimate your ancestry composition, flag certain health-related variants, tell you whether you're likely to have wet or dry earwax, whether you can taste certain bitter compounds, whether you're a carrier for conditions like cystic fibrosis or sickle cell disease. It's a strange and remarkable thing. The entire arc from Miescher scraping pus off bandages in 1869 to you casually checking your genetic ancestry on a phone app. But the real revolution, the one that's still unfolding right now, isn't just about reading DNA. It's about writing it, editing it, changing the code in living organisms with a precision that would have seemed like science fiction even 20 years ago. And the tool that made this possible has a name you've almost certainly heard: CRISPR. The story of CRISPR is one of those wonderful cases where a discovery in basic science, something nobody thought had any practical application at all, turned out to change everything. It starts in 1987 in Japan with a microbiologist named Yoshizumi Ishino, who was studying a particular gene in E. coli. While sequencing the region around this gene, he noticed something odd in the bacterial DNA, a series of short, palindromic sequences that repeated over and over, separated by unique spacer sequences. He noted it in his paper, didn't know what to make of it, and moved on. Over the next decade or so, other researchers found similar patterns in other bacteria and in archaea. In 2002, these repeating structures got a formal name: clustered regularly interspaced short palindromic repeats. CRISPR. But naming them didn't explain what they were for. The breakthrough came in 2005, when several groups independently noticed something remarkable about those spacer sequences, the unique bits between the repeats. When they ran those spacers through databases, they found that many of them matched the DNA of viruses known to infect bacteria. Think about what that means. The bacteria were carrying little snippets of viral DNA in their own genome, filed away between those repeating sequences, like a library of mugshots. And in 2007, researchers at a Danish food science company called Danisco, they made yogurt cultures, demonstrated experimentally that this was indeed an immune system. When a bacterium survived a viral attack, it could grab a piece of the virus's DNA and store it in its CRISPR array. If the same virus attacked again, the bacterium could use that stored sequence to recognize the invader and destroy it. Bacteria had adaptive immunity. Nobody had expected that. The mechanism works through a protein called Cas9, which acts like molecular scissors. The bacterium transcribes the stored viral sequence into a short piece of RNA, called a guide RNA, which then pairs up with the Cas9 protein. The guide RNA leads Cas9 to any matching DNA sequence, and Cas9 cuts both strands of the double helix right there. It's targeted destruction. Find this specific sequence and cut it. In nature, this chops up invading viral DNA. But in 2012, Jennifer Doudna at UC Berkeley and Emmanuel Charpentier, then at Umeå University in Sweden, published a paper that changed the trajectory of biology. They showed that you could reprogram the guide RNA to target essentially any DNA sequence you wanted. You could design a guide RNA in the lab, pair it with Cas9, introduce them into a cell, and cut the genome at precisely the location you chose. And when the cell tries to repair that cut, you can manipulate the repair process to either disable a gene or insert a new sequence altogether. Doudna and Charpentier shared the Nobel Prize in Chemistry in 2020 for this work, and it was one of those rare Nobel Prizes where basically everyone agreed it was deserved. Now, what makes CRISPR so revolutionary isn't just that it works. Scientists had other gene editing tools before, like zinc finger nucleuses and talons. What makes CRISPR transformative is that it's cheap, fast, and remarkably easy to use. A graduate student can design and order a custom guide RNA in a day or two for maybe $65. Within a week, they can have edited cells growing in a dish. That democratization of gene editing is what changed everything. Before CRISPR, gene editing was like hiring a master locksmith who charges a fortune and takes months to craft a single key. After CRISPR, it was like someone handed out universal lockpicks to every biology lab on the planet. And that's thrilling, but it also raises a question that should make you pause. If it's that easy to edit a genome, whose genome are we talking about editing? Because there's a crucial distinction here, and it's one worth sitting with for a moment. When scientists use CRISPR to edit cells in a dish, say, to study what a particular gene does, or to modify bacteria so they produce insulin, or to alter a patient's immune cells outside their body and then put them back, that's called somatic editing. You're changing cells that belong to one individual, and those changes die with that individual. They don't get passed on. It's like editing a single printed copy of a book. The original manuscript in the vault stays the same. But there's another kind of editing. If you edit an embryo, a very early stage embryo, just a handful of cells, or if you edit egg cells or sperm cells, you're making changes to the germline. And germline changes get inherited. They pass to children, and to their children, and onward through every generation that follows. You're not editing a copy anymore. You're editing the manuscript itself. And that distinction is where the conversation gets really, really complicated. Let's talk about what happened in November of 2018, because it sent shockwaves through the scientific world in a way that almost nothing else has in recent memory. A Chinese biophysicist named He Jiankui announced that he had used CRISPR to edit human embryos that were then implanted and carried to term. Twin girls, known by the pseudonyms Lulu and Nana, had been born. He claimed he had disabled a gene called CCR5, which encodes a protein that HIV uses to enter immune cells. His stated goal was to make the children resistant to HIV infection. Their father was HIV positive, and He Jiankui framed this as a medical intervention. The response from the scientific community was immediate and overwhelmingly negative. Not because the idea of preventing HIV is bad. Obviously, preventing HIV would be wonderful. But because almost everything about how He Jiankui went about it was reckless. For starters, there were already safe, effective ways to prevent HIV transmission from an HIV-positive father to his children through standard-assisted reproduction techniques. The medical justification was paper-thin. More importantly, the editing itself was sloppy. Analysis of the embryos suggested the CRISPR cuts hadn't been perfectly clean. There were signs of mosaicism, meaning some cells in the embryos had the edit and others didn't, which is a known risk with embryo editing and one that He Jiankui should have understood. There were also potential off-target effects, places where Cas9 might have cut DNA at locations other than the intended site. And here's the thing about off-target cuts in an embryo. Every cell that embryo becomes carries those unintended mutations. Every cell in the body. Every cell they might pass to their children. The scientific community had been discussing the ethics of germline editing for years. There had been international summits. There were calls for moratoriums. The broad consensus was that the technology was not yet safe or well understood enough to use in human embryos destined for implantation. He Jiankui knew this. He had attended these conferences. He went ahead anyway, apparently in secret, with what many scientists described as inadequate informed consent from the parents and insufficient oversight. Chinese authorities investigated and ultimately sentenced him to 3 years in prison for illegal medical practice. He was released in 2022, but the genie, as they say, doesn't go back in the bottle. The children exist. They carry edited genomes. And the world had to reckon with the fact that the first germline edited humans had been born not through some careful, internationally supervised process, but through the unilateral decision of one scientist operating outside the norms of his field. It was a sobering moment, and it highlighted something important about CRISPR's democratization. The same accessibility that makes it a powerful research tool also makes it harder to control. Now, let's step back from the drama for a moment and think about the technical challenges, because they matter enormously for understanding why most scientists said it was too soon. CRISPR is remarkably precise compared to earlier tools, but it's not perfect. Off-target effects are a real concern. Cas9 can sometimes bind to and cut DNA sequences that are similar but not identical to the intended target. In a research setting, you can screen for off-target cuts and discard cells that have them. In a human embryo, the stakes are entirely different. You'd need to be extraordinarily confident that the only changes you made were the ones you intended. And as of right now, we're not there yet. The tools are getting better. Newer versions of Cas9 and alternative enzymes like Cas12 and base editors that can make single-letter changes without cutting both strands are improving precision. But improving and perfect are very different words when you're talking about a person's genome. There's also the question of what we understand well enough to edit. Single gene disorders, conditions caused by a clear, well-characterized mutation in one gene, are the most straightforward candidates. Sickle cell disease, for example. We know exactly which mutation causes it. A single nucleotide change in the gene for beta-globin, the A to T substitution, at a specific position that causes the hemoglobin protein to misfold and the red blood cells to sickle. And in fact, CRISPR-based therapies for sickle cell disease have been among the first to reach patients. In December 2023, the FDA approved a therapy called Casgevy, developed by Vertex Pharmaceuticals and CRISPR Therapeutics, which uses CRISPR to edit a patient's own blood stem cells outside the body. They modify a gene to reactivate fetal hemoglobin production, a form of hemoglobin that doesn't sickle, and then infuse those edited cells back into the patient. It's a somatic therapy. It changes the patient's blood cells but doesn't affect their germline. And early results have been remarkable. Patients who suffered frequent agonizing pain crises have gone months and years without them. That's the hopeful side of this technology, and it's genuinely extraordinary. Think about what it means. Sickle cell disease affects millions of people worldwide, disproportionately in sub-Saharan Africa and among people of African descent. It's been understood at the molecular level since Linus Pauling's work in 1949, when he showed it was a molecular disease, one of the first ever identified. We've known the exact mutation since Vernon Ingram pinpointed it in 1956. And for decades, knowing the cause didn't translate into a cure. Now it's starting to. That arc from Pauling's molecular insight to CRISPR-based treatment spans about 75 years, and it traces almost the entire story we've been following tonight, from understanding DNA's structure, to reading it, to finally being able to rewrite it. But sickle cell is, in a sense, an easy case. One gene, one mutation, one clear mechanism. Most human traits and most diseases aren't like that. When you start talking about conditions influenced by hundreds or thousands of genetic variants, each contributing a tiny effect, things like heart disease, diabetes, schizophrenia, intelligence, height, the picture gets enormously more complex. Remember how we talked about the genome being far more intricate than a simple gene counting exercise? How 20,000 genes produce hundreds of thousands of proteins through alternative splicing, and how vast stretches of non-coding DNA regulate when and where those genes are active? That complexity means that for most traits, there's no single edit you could make. There's no one switch to flip. The idea that we could use CRISPR to make someone taller or smarter is, at least for now, far more science fiction than science. The biology is just too distributed, too interconnected, too context-dependent. And yet the conversation about enhancement, about using gene editing not to treat disease, but to improve on what's already healthy, keeps coming up. It's worth thinking about honestly, even if the technology isn't there yet, because the ethical frameworks we build now will shape how these tools are used when they do become more powerful. And they will become more powerful. That's the trajectory of every technology we've discussed tonight, from Sanger's painstaking manual sequencing to machines that can read a whole genome in hours. The tools improve, the costs drop, the capabilities expand. So where does that leave us? Well, it leaves us in a world where DNA is no longer just something we study. It's something we read, something we write, something we edit. The Human Genome Project gave us the reference text. CRISPR gave us the editing tools, and a whole constellation of other technologies, gene drives that can spread edited genes through wild populations, synthetic biology that designs entirely new genetic circuits, mRNA, vaccines like the ones developed against COVID-19 that use our own cellular machinery to produce protective proteins. All of these are branches growing from the same trunk of understanding that started with Miescher scraping pus off surgical bandages in 1869. Let's talk about one more piece of this puzzle, because it connects everything we've covered in a way that I think is genuinely beautiful. It's about reading DNA, not just from living things, but from the dead. Ancient DNA, the field that lets us sequence genomes from organisms that died thousands, even hundreds of thousands of years ago. And the person at the center of this story is a Swedish biologist named Svante Pääbo, who spent decades figuring out how to extract and read DNA from ancient bones and teeth, work that seemed almost impossible when he started, because DNA degrades after death. It breaks into smaller and smaller fragments. It gets contaminated with DNA from bacteria, from fungi, from every archaeologist who ever touched the specimen. The challenges were enormous, And for a long time, most serious scientists thought Pabo was chasing a fantasy. When he started this work in the early 1980s as a graduate student in Uppsala, he was actually supposed to be studying Egyptology-related immunology, but he became obsessed with the idea of extracting DNA from mummies. He did it in secret at first, sneaking into the lab on nights and weekends because he was worried his advisor would think he'd lost his mind. And honestly, the early results were messy. He managed to clone what he believed was DNA from a 2,400-year-old Egyptian mummy in 1985, and the paper got a lot of attention, but looking back, even Pabo himself has acknowledged that some of those early results were probably contaminated with modern human DNA. That's the fundamental curse of ancient DNA work. You're trying to recover tiny, degraded fragments of genetic material from specimens that have been handled by dozens of people, stored in museums for decades or centuries, colonized by microorganisms. The signal you're looking for is buried under layers and layers of noise. But Pabo kept at it. He moved to Berkeley, then to Munich, and eventually to the Max Planck Institute for Evolutionary Anthropology in Leipzig, where he built what became the world's premier ancient DNA laboratory. And the key breakthroughs came from a combination of extreme laboratory discipline and clever molecular techniques. The lab discipline part is almost comical in its intensity. Clean rooms with positive air pressure, full body suits, UV irradiation of all surfaces, multiple rounds of bleach treatment for every bone sample, drilling into the interior of specimens to avoid the contaminated outer layers. Workers had their own DNA sequenced so they could recognize and filter out any contamination from themselves. Every result had to be independently replicated in a separate clean room facility. It was forensic-level rigor applied to paleontology, and it had to be, because without those precautions, you'd just be sequencing the DNA of whoever cataloged the specimen in 1923. The molecular techniques mattered just as much. Remember how we talked about PCR, Kary-Mullis's polymerase chain reaction? That was both a blessing and a curse for ancient DNA work. It was a blessing because PCR can amplify vanishingly small amounts of DNA. Exactly what you need when your starting material is a few hundred degraded fragments from a Neanderthal bone. But it was a curse because PCR amplifies everything, including contaminants. If there's even a single molecule of modern human DNA in your sample, PCR will cheerfully copy that too. And since modern DNA is intact while ancient DNA is shattered, the contaminant often wins. So Pabo's team developed increasingly sophisticated ways to distinguish genuine ancient sequences from modern contamination, looking at characteristic patterns of chemical damage that accumulate in DNA after death, particularly a type of change where cytosine gradually converts to uracil over thousands of years. That damage pattern became a kind of authentication signature. If your sequences showed the right pattern of C to U changes at the ends of fragments, you could be more confident they were genuinely ancient. The first really big result came in 1997 when Pabo's team published a short stretch of mitochondrial DNA from the original Neanderthal specimen, the very bones that had been found in the Neander Valley in Germany back in 1856. Mitochondrial DNA, not the nuclear DNA that carries most of your genes, but the small circular genome inside your mitochondria, your cell's energy-producing organelles. Mitochondrial DNA is useful for ancient work because there are hundreds or thousands of copies per cell compared to just 2 copies of nuclear DNA, so you're much more likely to recover it from degraded specimens. And what that 1997 sequence showed was striking. The Neanderthal mitochondrial DNA fell well outside the range of variation seen in any living human population. It was clearly different. related, but different, which told us something profound. Neanderthals were not just ancient Europeans with heavy brows. They were a genuinely distinct lineage. But the real blockbuster came later. In 2010, Pabo's team published a draft of the Neanderthal nuclear genome. The whole thing, or most of it, pieced together from multiple bone fragments found in Vindija Cave in Croatia. And here's where the story gets really beautiful. Because what they found overturned a debate that had been raging for decades. The question was simple. Did modern humans and Neanderthals interbreed? The mitochondrial DNA had suggested no, because Neanderthal mitochondrial sequences aren't found in any living people. But the nuclear genome told a different story. When Pabo's team compared the Neanderthal genome to genomes from people around the world, they found that non-African populations carry roughly 1 to 4% Neanderthal DNA. People of European and Asian descent have small but measurable stretches of DNA that came from Neanderthals, inherited from interbreeding events that happened roughly 50,000 to 60,000 years ago, when modern humans migrating out of Africa encountered Neanderthal populations in the Middle East and Europe. Think about what that means. Somewhere in the deep past, your ancestors, if you have any non-African heritage, met Neanderthals, and they didn't just coexist. They had children together, and some of those hybrid children survived and had children of their own, and the Neanderthal DNA got passed down, generation after generation, diluted a little more each time, but never completely lost, all the way to you. You might be carrying Neanderthal gene variants right now that affect your immune system, your skin, your hair, your susceptibility to certain diseases. There's evidence that some Neanderthal gene variants helped modern humans adapt to the colder climates of Europe and Asia. They'd already been living there for hundreds of thousands of years, after all, and had evolved adaptations to those environments. When our ancestors interbred with them, they picked up some of those useful variants as a kind of evolutionary shortcut. It's called adaptive introgression, and it's one of the most fascinating things in modern genetics. And Pabo didn't stop with Neanderthals. In 2010, the same year as the Neanderthal genome paper, his team published something even more unexpected. From a tiny finger bone found in Denisova Cave in Siberia, a fragment so small you could hold it on your fingertip, they extracted and sequenced DNA that belonged to neither modern humans nor Neanderthals. It was a previously unknown type of archaic human, now called the Denisovans. We know almost nothing about what they looked like, because we have so few fossils, just that finger bone, a couple of teeth, and a jawbone found in Tibet. But their DNA tells us they were widespread across Asia, and that they too interbred with modern humans. People of Melanesian and Australian Aboriginal descent carry up to about 5 or 6% Denisovan DNA. One of the most remarkable findings is that Tibetans carry a Denisovan gene variant called EPAS1 that helps them tolerate high altitude, a gene they likely inherited from Denisovans, who had adapted to the high-altitude environments of the Tibetan Plateau long before modern humans arrived there. So, here's the picture that emerges. The story of human evolution isn't a neat family tree with clean branches. It's more like a braided river, with different streams of humanity flowing alongside each other for hundreds of thousands of years, sometimes merging, sometimes separating, exchanging genes along the way. And we only know this because of DNA. The fossil record alone could never have told us this story. You can't look at a bone and see that its owner had a Denisovan great-great-grandmother, but the genome remembers. Pabo won the Nobel Prize in Physiology or Medicine in 2022 for this work, and it felt like the culmination of everything we've been talking about tonight. Because ancient DNA analysis depends on every single breakthrough in the chain. Miescher's discovery of nucleic acids, the understanding of DNA's structure, the development of sequencing technology, PCR, the computational tools built during the Human Genome Project. Without any one of those steps, Pabo's work would have been impossible. It's all connected. And that connectedness is maybe the most important thing to take away from all of this. The story of DNA isn't really a story about one molecule. It's a story about how knowledge builds, slowly, unevenly, with wrong turns and dead ends and moments of breathtaking clarity. Miescher had no idea what his nucleon did. Levine got the structure wrong. Avery was right about DNA being the transforming principle, but couldn't convince the skeptics. Franklin died before her contribution was fully recognized. The genetic code took a decade of painstaking experiments to crack. The Human Genome Project revealed that we have fewer genes than a grape, which was humbling in the best possible way. And CRISPR, a bacterial immune system that nobody paid attention to for years, turned out to be the most powerful genetic engineering tool ever discovered. Every step in this story involved someone looking at something everyone else had overlooked and asking, what if this is important? What if this weird sticky substance from white blood cells is the key to heredity? What if this pattern of repeating sequences in bacterial genomes actually does something? What if we could read the DNA of someone who died 40,000 years ago? The answers to those questions changed everything. And here's what I find most comforting about all of this, especially at this hour, when things are quiet and the world feels a little simpler than it does during the day. DNA is the thread that connects every living thing on Earth. The genetic code is essentially universal. The same codons that specify amino acids in your cells specify the same amino acids in a bacterium, a redwood tree, a blue whale, a mushroom. That universality tells us something profound about the history of life. It means we all descend from a common ancestor, some ancient cell that lived billions of years ago, and the basic molecular machinery that cell used to read its genes has been conserved ever since. The details have been elaborated enormously. Gene regulation has become fantastically complex. Genomes have grown and shrunk and rearranged themselves in every way imaginable. But the core system, DNA to RNA to protein, has been running continuously for something like 3.5 to 4 billion years. It's the oldest technology on Earth, and it's running inside you right now, in every one of your roughly 37 trillion cells, as you lie there listening to this. Your cells are reading your DNA at this very moment. Genes are being transcribed into messenger RNA. Ribosomes are translating those messages into proteins. Those proteins are doing the thousands of jobs that keep you alive, repairing damage, fighting infections, consolidating memories from your day, regulating the slow rhythmic processes of sleep. You don't have to think about any of it. It just happens, the way it's been happening in living things since long before anything on Earth had a brain to think with. There's something deeply peaceful about that, I think. The machinery of life is ancient and reliable and astonishingly elegant, and it doesn't need your conscious attention to work. It was running before you were born. It'll run while you sleep tonight, and it connects you to every other living thing that has ever existed on this planet. The same 4 bases, adenine, thymine, guanine, cytosine, arranged in different orders, spelling out different instructions but always in the same language. One language for all of life. So let that settle in. Let the details we've talked about tonight drift where they will, Some will stick, some won't, and that's perfectly fine. The names, the dates, the mechanisms, they'll be there if you want to revisit them. But the big picture is simple and beautiful. A molecule that carries information, a code that translates it, and a system so robust that it's been working without interruption for longer than mountains have existed. You're part of that unbroken chain. Every cell in your body is a small testament to billions of years of accumulated ingenuity, most of it arrived at not by any conscious designer, but by the patient, blind, astonishing process of evolution. Variation, selection, inheritance, repeat. And now your own cells are doing what they do best in these quiet hours. Repairing, restoring, consolidating. You don't need to follow the thread any further tonight. The story of DNA will still be there in the morning, still unfolding, still being written, in laboratories and hospitals and caves and computational servers all over the world. But for now, this is a good place to rest. The molecule that makes you who you are is taking care of things. It's been doing this for a very, very long time. It knows what it's doing. So you can let go and drift and sleep